Now, the next important biomolecule which you need to cover is the nucleic acid. Now, what are nucleic acids? They could be DNA or RNA. What is the basic structure of these two? What does the DNA consist of? For example, 4 kinds of bases joined to a sugar phosphate backbone. What are these bases? Their structures that you need to know. Then what is a nucleoside and what is a nucleotide? Which sugar is present in a DNA and which is present in RNA? Then what are the 4 bases present in RNA? Then who discovered the double helical form of DNA? Then what are the important features of Watson and Crick's DNA model? For example, the 2 helical polynucleotide chains are coiled around a common axis. Then the purine and pyrimidine bases are on the inside of the helix, whereas the phosphate and deoxyribose units are on the outside. Then what is the diameter of the helix? It's around 20 angstrom. Then the adjacent bases are separated by what distance? That is 3.4 angstrom, and the rotation is of 36 degrees. Then how many residues are there in each turn? There are 10 residues. And the other features which he emphasized was the 2 chains are held together by hydrogen bonds between pairs of bases, and then the sequence of bases along the polynucleotide chain is not restricted in any way. And then what is the most important aspect of the DNA double helix is the specificity of the pairing of bases. Watson and Crick deduced that the adenine must pair with thymine and the guanine with cytosine. After that, you should study about the DNA replication. What are the important features? For example, the complementary chains act as templates for each other in DNA replication, and the replication is semi-conservative. We should know what is semi-conservativeness of DNA replication. What are the other important features of DNA double helix? That is, they can be reversibly melted. DNA molecules are generally very long molecules, and you should know the length of DNA molecules in various organisms. And the DNA molecules could be circular, supercoiled, or linear. Then how does the DNA replication happen? What are the enzymes involved in it? And what are the different steps? What is the role of DNA polymerase? And what are the components which it requires for the synthesis of a DNA chain? DNA molecules are generally double-stranded, but during part of life cycle of some viruses, they are single-stranded. So you should know about them. What is the role of them in these viruses? And how does the replication happens? You should also study about some viruses where RNA is the genetic material. Then how does the replication occurs in them? For example, RNA tumor viruses and other retroviruses replicate through double helical DNA intermediates. You need to study about them in detail. So according to the syllabus, you need to know about few important proteins, their structures and functions, like myoglobin and hemoglobin. They are the mainly oxygen carrying molecules, but the binding of oxygen to hemoglobin is regulated by various factors. You need to know what are these factors, and the interaction between spatially distinct sites termed as allosteric interactions are responsible. Need to know the structure and function of these molecules, like you should know where does the oxygen binds on the hemoglobin, that is the heme prosthetic group. You should know about the structure of this. Then what is the structure of myoglobin? That is a compact structure and very high content of alpha helices. Then what are the important structural features which are important for the function of the proteins? Like the hindered heme environment is essential for reversible oxygenation, and also the carbon monoxide binding is diminished by the presence of the distal histidine. Now, after myoglobin, you need to study the structure of hemoglobin, which consists of 4 polypeptide chains. What are these chains? They are like 2 alpha and 2 beta chains. And how the hemoglobins of embryos and fetuses are different from that of the adult. And the structure of hemoglobin subunits are resembling the structure of myoglobin. Then what is the role of allosteric interaction in coordinating the transport of oxygen, carbon dioxide and protons? This is the major difference between hemoglobin and myoglobin, that hemoglobin is an electrostatic protein and while myoglobin is not. So how are these differences expressed? For example, the binding of oxygen to hemoglobin enhances the binding of additional oxygen to the same hemoglobin molecule. And the affinity of hemoglobin or oxygen for oxygen depends on pH, whereas that of myoglobin is independent of pH. And the oxygen affinity of hemoglobin is further regulated by organic phosphates such as 2,3-bis phosphoglycerate. So you should study in detail how the oxygen binds cooperatively to the hemoglobin. What is the graphical representation for that? What is the Hill coefficient? And for what is it used to measure? Then how does the cooperative binding of oxygen makes hemoglobin a more efficient oxygen transporter? Then what is the Bohr effect? How does the BPG affects the oxygen affinity of hemoglobin? Then how does the fetal hemoglobin has a higher oxygen affinity than maternal hemoglobin? Then how does the oxygen binding switches quaternary structure by moving iron into the plane of the porphyrin? Then how does carbon dioxide lowers the oxygen affinity of hemoglobin? Then what is a sequential model for allosteric interaction subunits change conformation one at a time? Then what is the concerted model for allosteric interactions? What is sickle cell anemia? How does it affect the oxygen binding? And what is the cause of sickle cell anemia at the molecular level? A single amino acid in the beta chain is mutated. And how the high incidence of the sickle cell gene is related to the protection conferred against malaria. Then how the fetal DNA can be analyzed for the presence of the sickle cell gene. Then what are the other molecular pathology related to hemoglobin? You should know about them. For example, thalassemias are genetic disorders of hemoglobin synthesis. So these were the important points which you need to go through about myoglobin and hemoglobin. Then after that you need to cover a few important enzymes like lysozyme, ribonuclease, carboxypeptidases and chymotrypsin. First of all about lysozyme, who discovered lysozyme. Then what is the function of lysozyme? That it cleaves bacterial cell walls. You should know about the basic structure of lysozyme. That is a compact protein with a complex folding. Then where is the active site located of the enzyme? Then what is the mode of binding of tri-N-acetyl glucosamine? That is a competitive inhibitor. Then you should know about few points regarding the structure which leads to the enzymatic mechanism, like how would a substrate bind. Then which bond is cleaved? Then which groups on the enzyme participate in catalysis? Then what are the different factors which are critical for catalysis? For example, one is the carbonium ion intermediate. So after lysozyme, you need to study about ribonuclease A. What is the structure of ribonuclease A? And then what is the function of ribonuclease A? What is the mechanism? For example, a cyclic phosphate intermediate is formed in the hydrolysis of RNA by ribonuclease A. So you need to study this in detail, how the phosphorus is pentavalent in the transition state for RNA hydrolysis. And what are the residues which play critical role in catalysis? And go through the mechanism in detail. Then after that you need to study about carboxypeptidase A, which is a proteolytic enzyme. Need to know first of all the structure of the enzyme, and it is a zinc containing enzyme. Then what are the important features for the catalytic mechanism of this enzyme? First is an induced fizz, then other one is the activation of water. What is the effect of substrate binding on the enzyme? There is a large structural change. Then what is the role of zinc ion at the active site, which activates a water molecule? Then another important enzyme which you need to go through is the chymotrypsin, which is a serine protease. The important part of the enzymatic mechanism is that a part of the substrate is covalently bound to chymotrypsin during catalysis. The acyl group is attached to an unusually reactive serine residue on the enzyme. Then what is the catalytic role of histidine 57 and how was it demonstrated by affinity labeling? Then what are the roles of serine, histidine and aspartate in chymotrypsin? They form a catalytic triad. Then what is formed as the intermediate during the catalysis by chymotrypsin? It is a transient tetrahedral structure. Apart from this, there are many other important catalytic enzymes like trypsin elastase, which are variations on the chymotrypsin theme. Then there are other classes of proteolytic enzymes like serine, zinc, thiol and aspartyl proteases. So go through them with examples. So these were the few important proteins for which you need to study the folding and function in detail. And that was all about biomolecules. And you need to go through these thoroughly because these are the basics of various studies and various other fields. And a lot of questions are being asked from here. And the time which you will need to prepare is around 3 to 4 days. I hope the information which I share is going to be useful for you, and I wish you good luck. Thank you.